That was just a reminder. I don't know, do we have the recording? I think the recording's on again, and email list. If you could send that out, that would be great. So this is just a picture of us shaking up a bunch of packages, and we had a really big kind of field day with lots of students needing a lot of things. And then this week we also did a sugar roll on the entire hive. So I'll show you how that turned out. And first thing Randall said was, why? This is, oh, I'm going to try to mute this. You don't need to hear Sheldon. He's yelling at all of us to do things, shake the frames in there. So what Dustin in the background and Luke in the background are doing are finding the queens beforehand. We put them aside, and then we're basically shaking the bees off these frames into a funnel that then gathers them into this plastic tub. And the tub has air holes in it, and it's just basically collecting all the worker bees. And then Rogan over here on the left is collecting the honey frames because he's using those in an experiment to look at how resources from a good location versus resources from a poor location affect queen rearing capacity. So he's setting up a bunch of queenless nukes, and we're taking resources from this location and comparing that with our bee kill site and looking at productivity. So that's us kind of shaking the bees into this big old tub, and we'll just continue to do this. I usually will shake 2 to 3 colonies worth of bees just to get them equalized and randomized enough so that all the packages will have kind of a random assortment of worker, nurse bees, foragers, etc. So then the first time we did this in the spring, we had the packages on the scale. We were just kind of scooping in until the package showed 2 pounds. But the bees didn't like it. It was kind of a mess. The scale was teeter-tottering, so it was a little bit hard to read and see. So we kind of modified that method a little bit this year with just basically, we took 1 measure of 2 pounds of bees and used a cut up, what is it, this milk jug? So to measure out 2 pounds or 3 pounds of bees, and we just draw a line so we can just scoop and toss rather than measure them out each time. Because it was taking a lot of time to sort through each package and measure them out, but this was an easy way for us to kind of make 2 pound packages a little bit more quickly. So in this first one, I'm just marking where the 2 pound bee pile would sit. And then I like the jugs because they've got a nice built-in handle. And the smaller quart jug was actually better for some of the smaller packages because the bees just went up to the top and I was able to stick the whole entrance into the package and just dump them quickly. But it's a little easier. You can see that they're a little bit less roused up and jazzed and mad. And then the sugar can. Let's see. So that day we inspected 24 hives, frame to frame, and made up of those 36 nukes, and they were all queenless because again, Rogan is interested in looking at how food resources from one bad location versus good location affects queen rearing capacity. And we have a site that is experiencing bee kills and consistent bee kills. So we're taking those resources and trying to see if those bees can recover if they lose a queen. And then a lot of the packages, the 9 packages and these hives were transported to Kimmel so that we could put our Great Plains Master Beekeeping Training Day. This is just a test out day. We still have COVID restrictions. So everybody had to, this is from invitation. So the board members or the governing committee of our group, the masters, the Great Plains Master Beekeeping Program governing committee is made up of board members of the associations, basically. So Nebraska Beekeepers Association, Northeast Kansas beekeepers, Kansas honey producers, Missouri beekeepers, Omaha beekeepers. So a lot of these folks on the board sent out representative folks that are already teaching classes that would be really good for helping us proctor some of these exams in their own states. So we had them come, they drove to Kimmel and did their test out so they could be certified as masters, or they can go into the level that they need to, so they can start providing mentorships and offer tests themselves. So this is, I think, Megan Vedder with MBA. We've got Becky and Christy from Kansas. And then I think we also had Buzz come out as well. I didn't have anyone from Iowa come out, but we are trying to have some more folks come out and join us for that. So ideally what we're trying to do is have them see how this field exam works out so that they can proctor them in their own states. And our program is a little different than a lot of the other programs because we're not a bee college, we're not a bee school where we're just offering a lot of virtual content. We're trying to help beekeepers with the field applied knowledge, with the practical applications of the management. So we do some lectures, but a lot of it is focused on, okay, let's improve your management skills. Let's teach you some of the things that you need to do in our area to deal with the pests or to deal with certain chemical stressors and everything else. We don't sell any of these bees. These are only for research packages. And then we do a lot of crazy things because it requires a lot of things. So this is sugar rolling the entire hive. And I bring this to people's point because they talk about, you know, sugar rolling hives as an effective varroa treatment. And that is dumping sugar in the hive. And that doesn't work because what you're doing is you're dumping the sugar on the hive, but it just goes down and it's not causing them to heat up and to generate heat. When we sugar roll a hive, we're actually taking all the bees, just like when we shake packages, we find the queen, we shake all the bees into the box, we powder sugar them, and then Dustin or somebody else who is stronger takes the box, turns it upside down and shakes the gajibis out of it. So let it sit for a few minutes. Huh?
Question, where did you just make that box? Did you have that made? Or is that something?
Yeah, so actually that I inherited like a dozen of those boxes because Marion was doing OA treatment trials and doing a bunch of sugar roll trials. So on the side, he had it built, but on the side, there's gates that lift so you can dump the bees out, and then the screen can open and close. So it's a nice handy little box. But you guys see all the mites that were on there? So that's the way that we can get a nice quick sampling of varroa mites for our colleagues. They're looking at like they're looking at what are they? New varroicide products and certain receptors that varroa mites have. So they're looking at resistance receptors and possibly receptors for targeting new products for varroa specifically. So their interest is a little bit more lab-based. And so we just give them the mites to play with. And then the bees get dumped in.
There's a question from Eric Paisley. How do you count the mites when it is that big? So I guess whenever you're picking for so many bees, how do you count mites?
We aren't actually counting the mites. So those mites aren't being quantified. We just we collect them all and then we freeze them. And then the lab-based people take the individual mites and put them in their experiments for looking at certain receptors. I don't actually know what they're doing with it, but they need live mites and they need to flash freeze those mites to look at their physiology. For us, we do a sugar roll and we just determine management through the sugar roll. But this is something that Sheldon is, I'm having Sheldon kind of look into whether or not he wants to compare, you know, when you sugar mite roll with a jar versus the entire colony versus just throwing sugar into the hive, what do you get? Does the sugar roll kill the mites? Not really. It just makes them hot. The heat makes them hot, and they get you know, they fall off their host. And then if there's nothing to glue them to a sticky board or to hold them down, they'll just crawl back up the frame or find another bee to get, you know, nice and cozy with. So that's why just pouring sugar right in the hive doesn't really do anything. If you were to pour sugar into the hive and then somehow put something like a sticky board to hold the mites that fall down, that would be one way. But I imagine that if the mites are falling, so is a lot of loose sugar falling. So eventually that sticky substance won't really be holding much of the bees down or the mites down. That's all I had in terms of videos. They still I mean, my phone died before I could finish the bee mound going down, but basically I go back and I kind of push them around with a hive tool. I pull out a frame and some of them fall in. It'll take them a while to clean up, but you know, they'll clean up.
And Judy, when I saw you do a sugar roll like that, a sugar shake, that's when I first learned the term ghost bees. Did I ever show you the video we put on Instagram with, we had Halloween music and we were shaking the ghost bees out?
Slow-mo.
Yeah, you kind of inspired that with all the little white ghost bees. Anyway.
They are so cute when they're all covered in sugar.
Yeah, they're not too happy either, but yeah.
This one gets a little bit tricky because depending on how hard you shake those bees off, if you get nectar in there or honey in there, it starts to get really sticky and then things get kind of wet and yeah. That's one thing you have to be careful about the sugar rolls. If you live somewhere humid, the sugar starts to like glue to the glass jars and you know the mites can stick to the sugar. You got to clean it out more often.
Judy, did you answer the question in the chat? Does the sugar roll kill the mites or just make them loose of the bees?
Yeah, I answered Mark's question about no, the sugar isn't going to kill the mites. It's just the sugar irritates the bees generate the heat dislodges the mites, then the beekeeper kills the mites. That's pretty much the progression of how that works. Yeah, the bees are really just warming up and the mites get off. And that's what I was saying is if the mites don't have anything to hold them down, to glue them up, or to remove them, they'll just climb back and find a new host. And then Shauna said she watched a live meeting last week from some scientific beekeeping thing, and they tested a bunch of different sampling and measured their heat with heat guns. And one other method results where the bees' temperatures did not rise at all. Yeah, and I guess I would have to see what they were talking about because it's going to depend on what type of heat gun they're using, how far away they're doing it, and what methods they're talking about. Certainly, you know, if you're just heating it up in the hive, they may not generate more heat than you think they would. But if they're confined in a jar or confined in a box, like the shaker box, they're going to generate more heat than they would just naturally in the hive when you throw powdered sugar on them. Anybody else have questions or pictures or videos to share? I have some non-bee stuff, but we found a bat in our backyard or in our garage. It was kind of a proud mom entomologist moment this week because my 6-year-old helped identify, we have these little glass vases that hang on the wall for flowers. And she pointed out that we have mosquito larvae living in them. And then like 2 days later, she's like, I think the cat's got a mouse or a bat or something. It's got teeth. It's really kind of scary. And I go over there and yeah, she had a big old bat in her garage.
Have you ever, entomologist sidebar, have you ever experienced bat bugs? They look like bed bugs, but they are a little parasitic cousin of the bed bug that live off of bats. And I had a friend recently find them in their house where they also have quite a few bats. And they were worried it was bed bugs, but they pulled out a little USB microscope on their computer and they self-ID'd it as a bat bug instead.
Yeah, I've heard of them, but I have not seen them.
They looked almost identical. I hadn't seen one either. So anyway, I just thought that was cool. Maybe keep an eye out for that.
No, I had a short stint in like rehab in college. You know, I did wildlife rehab when I thought I wanted to be a vet like every other person. And the last time I had to rehab the bat still died, end up with rabies, and then I had to get rabies shots. So this time I was like very careful not to be touching the bat. And I was like, I'm not dealing with it. I'll give it to the bat experts. And I called the wildlife team to pick it up, or I dropped it off. It was no fun. It was also around Halloween. And so, me and my roommate, we had to go through a series of rabies shots in the month of October. They called us the Bat Girls. And Bat Girls are here for the rabies shots again. Okay, so Mike is asking a question about, is there a way to test pesticides or other toxic stuff in honey? Are there concerns on on what's in honey? And that's a great question. There actually is. There's a lot of concern that contaminated, nectar and pollen are going to make its way back to honey. One of the good things is, honey tends to be more liquid-philic, so hydrophilic, so it tends to be more water-based. And a lot of the chemicals, the agrochemicals, are more lipid-based. So they tend to secrete and hold into the wax. And so honey tends to be less contaminated than your wax and your pollen. But that's where the dead bee traps and all the discussion about maybe making sure you are able to recognize nurse bees in your hive is important. Because if you think about it, if the nectar is contaminated and really contaminated at a high level, those bees aren't going to make it back home. They have to consume that in order to carry it home. So that biases the honey to have lower residues in the first place coming in. And then the fact that their physical property allows them to not soak up some of those chemicals and instead leach it into the wax, that's also really good for the pollen or for the honey. Where I would be careful is pollen products, because the bees don't have to consume it to bring it back. You can have higher residue levels show up in your pollen. They just pack it on their legs and bring it back. So there's the potential there for higher residue levels. There are concerns. There's always concerns on people's health, but honey is one of those things that they do set food tolerance levels for pesticide residues in food products like honey, but luckily that's not one that has had problems with high contamination. Is it possible to have, is it possible when you have a super and a queen excluder on for a virgin queen to mate and come back on the top, and another virgin queen to mate and come back in the bottom, and two queens in the hive? Yeah. Yeah, anything's possible with honeybees. No,
we had a queen. We had a queen above and below the excluder this summer. Anything's possible.
Sometimes the virgins and also the newly mated queens are still thin and slim enough to go through those excluders. And then once they start laying, their abdomens swell up and their ovaries swell up, and then they can't go through. Yeah, and so if they're on either sides of the excluder, they don't even know.
Eric wants to know, Eric tried to harvest honey this year, ran into an issue where the honey would not spin out in the extractor. Had the cells open properly, but the honey was too thick. Is there something that causes this?
How cold was your room?
Was the honey crystallized?
Mm-hmm.
Yeah, Eric, feel free to unmute and
Working on it.
Oh, okay.
Sorry. So it was, how cold was the room? It was in a garage when it was 90 degrees outside. And I had pulled the supers maybe 40 minutes before I tried to spin it out. So they weren't really cooled off. Like they weren't cold. I was just shocked at how thick. Was it crystallized? I'm not sure. I don't think so. But to be fair, I'm not really sure if I would know if it was or wasn't.
What do you have around there? What kind of forage?
Let's see here. I back up to a. I'm in the middle of Omaha, but I do back up to a pretty large creek in a pretty large, or not a large forested area, but the creek is loaded with trees and lots of wild food, for lack of a better word.
This is Mark Welsh in Omaha also. I had a similar situation, not that I tried to spin honey out, but I was trying to get honey moved from 3 or 4 frames down into 3 or 4 other frames so that the cells would all be full and the bees would cap it if it was actually right. I turned 3 of the 4 frames in the top super above an inner cover upside down. And I went back 24 hours later and the bees hadn't started to move very much of it, if any, out, and none of it had dripped. So I think the honey is just super dry and thick, and it's not capped. You know, they continue to heat that stuff up to 90+ degrees inside the hive, and if it's not capped, it's just going to keep evaporating. That's my guess anyway. I'm going to find out tomorrow. I'm going to be extracting tomorrow.
So what would cause it to crystallize in the hive?
There are certain honeys that will crystallize. I'm sorry, there are some honey nectars that crystallize more easily. I don't know if there are any that will crystallize in the hive during the summer, because normally if you see crystallization, it's like over winter where the bees weren't over it and the honey just, got cold. I don't know if, like in Nebraska, it's been hot. There's no cold, there's not enough cold temps to crystallize your honey. This is this year's honey, right? This isn't leftover from last year?
No, it's this year.
This is Robert. The canola crop tends to have a higher glucose ratio, and it will crystallize readily in a short period of time. So I think it might depend on what the bees were foraging on. It might have been something that was higher in glucose. I'm not saying it was canola, but
that's fair.
Yeah, I think it's
less.
not the cold, it's not crystallization. Yeah, it's.
I mean, I probably left it in the extractor for a good 15-20 minutes. That thing spinning around trying to get stuff out of it, and it wouldn't move. I even had a frame break on me from the frame broke, but the honey still didn't come out. But
Yeah, we've seen honey before that's very like sticky and thick, almost tacky like. Can't remember where that was. I'm trying to remember what source of honey that could have been from. But yeah, there's certainly some plant sources or nectar sources that are going to have sugar compositions that are lower in moisture or contain lower moisture or have the different glucose sucrose ratios that causes crystallization. And then on the opposite end, there's some nectars that don't crystallize barely at all.
Yeah, the hard part I have is so I'm in the middle of Omaha, so it's a very urban style environment. Like I back up to a creek, but I mean, and there's a lot of trees and there's a lot of forage there, but it's not like there's a large canola field or a large corn field or a large, like a field nearby that I know they're eating on. It's hard to tell what they're eating.
Yeah, in urban areas, it's tough because there's flowers from shrubs and trees to consider as well, you know, a lot more so than your open landscapes where there's just a lot of wildflowers. So there's a lot of like ornamentals that might be providing nectar sources. If you're still having trouble, maybe try and turn on some, I know it's hot in the garage, but maybe try and turn on a little bit more warmth right on the combs and try to heat up that honey a little bit so it's a little bit more viscous. I don't know if anybody else has any other tips for them.
Thank you.
Yeah.
So I've got another question for Eric. Is a lot of your honey doing that, or was it just one frame or one super, one hive?
So last year's honey did not do that at all. This is my second year, and this was the first super that I harvested, and it did it. So I wasn't expecting it. This was the first time I ever used an extractor, and so I was kind of excited about it. And then I spun everything out, or I attempted to spin everything out, and I didn't get anything. And so it was kind of an interesting experience. So this is the first time it's happened. But to be fair, it's a small sample size.
And it was capped before you started.
Correct.
Very interesting.
Were you spinning fast enough?
It broke the frames, so I feel like it's going pretty good. And it spun for a while, so it was an electric extractor, so it was just going. Like you just set it and let it spin, and I ended up just like when it was all like I got some honey. I think I got about, I don't know, 4 to 5 pounds or so out of the super. So I got something out of it, and I ended up just scraping everything else off, just cleaned off all the frames, so there wasn't any more comb on it, and just stuck whatever was wet back in the hive and harvested the wax off of it just in case it was a wax issue, or I don't know. I just wasn't really sure what to do. Now the cool thing was that was 3 weeks ago or so, and I put that super back on the hive, and they've probably drawn out and filled half that super back up, which is kind of cool. So hopefully I will get another harvest, and hopefully it won't be of Play-Doh style honey, and it's actually useful honey.
Yeah.
So Jacob says, a couple times I run into what seems like fermenting honey. What could cause this, since honey without too much moisture should be almost resistant to it? Funny thing is, I don't notice it in the gallon glass jars I put honey in after harvesting, but when I pour it into a smaller container for handling, like a 5-pound plastic jug, suddenly it becomes more opaque with tiny bubbles, as if I disturbed something carbonated. Small jars filled to the brim with mediocre seals, wire ball, rubber gasket will ooze honey. Freezing seems to stop this process. Judy and Randall?
Well, I mean, I've seen like bubbling in frames with uncapped like nectar cells that are fermenting, and you know, the little bubbles that come out, and it's like indication that the sugars are turning into kind of the ethanol process. A lot of that is like driven by yeast, and so it sounds like freezing your frame or freezing that honey killed off those yeasts that are using the sugars to convert and ferment into, you know, I'm not entirely sure about pouring it from one jar to another and having those. I wonder if those bubbles you're talking about is the foam that, typically kind of shows up in honey. I'm not sure. Anybody with more experience with honey in jars? Kind of talk about the bubbles.
I just I got some jars at the Menards today so that I could give it to people that have kind of supported our project this year. And I guess you can't really see it. I had honey sitting on our shelf from our earlier harvest, where I bottled it pretty quickly after we harvested it, so it still had a little bit of settling out to do. But I scraped off the top, and then when I poured it into these smaller jars, yeah, I had some kind of like a bubbling action going on, but it's mostly gone away now.
I wonder if the bubbling action is what you're seeing is crystals, because when we're judging honey, and we're moving things around, sometimes it's hard to tell what's a crystal, when they're starting to form versus when they're fully formed. And sometimes it looks like opaque waves in your honey, like if you're pouring one with another, sometimes you'll see kind of cloudiness. And that's kind of small little tiny crystals that are starting to form and just got disrupted when you kind of move the honey. I don't know, Dustin or Randall, do you guys have any thoughts on that? I think that's what it is the opaqueness is tiny, tiny little crystals forming.
Yeah, I'm not sure.
I agree, though. I would try to get a refractometer, or at least talk to your local club and see if they have one that you can kind of just check your moisture content. Make
Yeah,
sure it's
we
not
don't,
fermenting.
but some members do. So yeah, they're around. And some of them are so cheap. Those might be the ones with unusable instructions.
Rick wanted to know if he doesn't harvest honey until October, is that too late? It kind of depends on where you are.
I'd be worried about the mites.
I always tell people it's a big risk the longer you leave it on. If something happens to your hive, you're going to lose all that honey.
And Rick, you're not one of the California beekeepers, right? Because your guys' time in season is totally different.
No, I'm from Unadilla.
Yeah, October seems like it's a little bit too late because, like we mentioned earlier, it's not about the mites that are on the bees going into winter. It's about cleaning up the mites before the last generation of brood is being reared. For the last brood and babies, you want to have a nice mite-free environment for those babies to rear up in a mite-free, non-vampire, mite-sucking environment where they can grow up to be nice and fat and robust for winter. So if you treat in October, you'll knock down all the mites on the back of the bees, nothing to protect them when they were developing in the comb. So they might be weaker, they might have a hard time, withstanding the winter.
Right. And two other reasons, like someone already mentioned, if you wait till October, and especially here, at least in Iowa, we don't have a lot of resources for the bees in September. So a lot of them will eat up everything that they've stored so far. So you'd be losing some of your honey that you would normally harvest. And then another reason for us here as well is the fact that in the late season, the only resource that's available is goldenrod, and that can really change the flavor of your honey to something that a lot of people don't like. So I would say those are the three reasons: mite control, avoiding goldenrod, and taking the honey before the bees can eat it would be three reasons to be thinking about doing it at late August, early September. Yeah.
Yeah, I personal experience in central Iowa, going from doing it after the Iowa State game in mid-September to doing it before Labor Day, our hive survival's gone way up. Overwintering's gone way up. Doing it just that 3 weeks sooner.
Okay, so if he doesn't want to extract until October, should I just pull these supers off and freeze them until he's ready? Because he's the one that's got the extractor. And go ahead and treat my hives then?
That would be one option, I think. Or you could talk with him and say, hey, can we extract some of it now and some of it later? Maybe just work out a deal with him. But one option would absolutely be to remove those pull those honey supers and freeze them. Judy, do you have any recommendations about freezing honey frames, like do's and don'ts?
Well, I mean, unfortunately, it's kind of a pain because if you freeze that honey frame, you're going to have to deep freeze it, unthaw it and warm it up before you extract it. So, I mean, if you have maybe a cooler, a walk-in cooler, you could just temporarily store them, prevent wax moths from getting them, and they won't be as tedious to warm them up. But yeah, it's kind of, 3-4 weeks is too long to let them sit. The wax moths will get into them and you'll get beetles and other pests. So if you're gonna pull them off, definitely store them somewhere where you're not gonna have little critters dealing with them. But I don't know, freezing them will also require you to, how much honey are we talking about? Maybe that's a better question.
I just have one super full, maybe a half of another one, but I'm also a home brewer, so I have a keezer that I could set it in too. So
Yeah.
that would work.
Yeah. Yeah, I think pulling it off and getting your treatments on early would be better. The other advantage is if you pull your honey off and they start collecting fall forage, you don't have to feed them as much, so there's a lot of advantages there. And if honey production isn't really your main focus, then survival, you probably would want to hedge your bet on survival than getting an extra couple of gallons of honey.
Yeah. Okay. Thank you.
Mm-hmm. Goldenrod equals gym socks. That's what I heard smartweed was like. And buckwheat is also kind of got a strong flavor, but I don't find it like as disgusting or repulsive as the gym sock taste. Are people opposed to buckwheat honey? The taste of that? Because there is interest in more cover crop research on buckwheat, but I didn't want to promote it if nobody wants it.
I like it.
Buckwheat is a really dark honey. It's got a much higher antioxidants, and it's a great cover crop, but people don't grow them out here.
It's very molasses-like, isn't it? Kind of that richer, deeper flavor.
Yeah. Okay, Mark says small hive beetle eggs and larvae need to be frozen before storing for later extraction. And then, let's see. Marcia was saying, have done an OAV treatment already due to seeing deformed wings. I'm still seeing mite drop on the removable bottom board. Can you go over best practice for doing OAV? I think with oxalic acid, the label says you can use it up to 4 times a year. That would mean if you've used it in the spring, count how many times you've used it. But that's part of the reason why Dustin and I, we try to rotate with OA and something like Apigard or something. Because if you need to use it in the spring and you need to do cleanup in the fall, you don't want to overuse the illegal number of times to apply it.
But so since oxalic acid won't go through capped brood, you're going to want to do at least 2 treatments, correct? In order to first get all the open brood in your first round, and then do a second treatment after all the other brood has been opened up.
Honestly, we use, and Dustin could talk more about this, but we use the Apigard kind of this time because there's a lot of brood. We're also playing around with the idea of trying some of that Hap-BEE product, the number 3 version. But we reserve the OA treatment for when they're almost broodless, at the late fall or early spring. So we kind of conserve the OA treatments to kind of that time of the season, and then things that are more effective for capped brood like thymol or something else.
So you guys don't ever use the formic acids, the My Way Quick Strips or Formic Pro?
We do, but it's temperature sensitive, so it's something that in the past when we've done it, that's been usually
It would,
early.
right. It would have been good to have on this week.
Yeah, yeah.
Yeah. Yeah, the first year I was here, I got a call about a pesticide kill, and I went out to inspect, and it was a yard of, I think, almost 130 bees, colonies, and there was maybe 12 left. And they were all dead outs, but it was really hot out, and it did look like there was a lot of brood burn and brood damage and queens lost, and it looked like it was more improper formic use than the pesticide use, because it, you know, like it was really hot, and the way they described it was, you know, these were one deep colonies, and they had put them on and then reapplied, and it was too much. And yeah, it was a hard lesson learned for these beekeepers.
What time of year was that? Can I ask? Like, how hot was
it? It was in June, but there was a hot streak, you know, and so they were treating when they didn't need to at a time, and they had just came back. I think it was actually end of May or June. They had just got back from some pollination contract and were plopping it down and adding a treatment. But these colonies weren't very big, and it was really too hot for that. Okay, and then let's see. Tony says, I have partially drawn frames with very little honey on this honey supers I pulled off to allow me to treat my hive. Can I store them over winter with paramoth and then put them back on the hive next year? What's very little?
This is actually a very great question, because we store our frames with resources in them, because we don't have anywhere else to put them. We don't have a big freezer or cooler or anything like that. And I mean, when it gets cold out, it's not something that we have to worry about too much. But once it gets warm and we have our bee barn is full of these hot, humid frames, I don't know what to do, because we have so much equipment from research from previous years that we didn't use this year. It's just, I don't know, maybe going bad.
Yeah, I mean, there's plenty of combs with honey and pollen that we store and we put paramoth on, but we also don't intend to like extract it for human consumption.
Yes.
So
yes.
I would be careful if you had a lot of honey in the supers, maybe just give it a quick spin out and then, you know, let the bees clean it out, the wet comb, and then store it as dry comb. You'll have less pest problems, and you won't worry too much about contaminating the honey that you might extract next year. It's hard to say if the bees are going to consume what's there or they're just going to add more nectar to what's there, in which case you don't want it to have, you know, residues remaining.
Barb says,
asked, go ahead, Randall.
I was just going to say, I was going to read Barb's comment for you. 28 days since she did a split with capped queen cells, still no sign of eggs. When should she just give up and recombine?
Yeah. Now. It doesn't seem like 28 days since you split, and
how big are
yeah, you still don't have eggs?
So I'm pretty sure this was a hive that had capped queen cells, but the queen's still in it. And so I pulled the queen off into a separate nuke. She's doing great in the separate nuke. I have no idea why they were building supersedure cells, because she's got a great laying pattern. So my instinct is it's time to just put them back together, but I didn't.
Yeah, it sounds like they're making supersedure cells, or they're drawing queen cells in the nuke. Is that what you're saying?
Know if.
Sorry, Barb, I couldn't hear you.
Sorry, yeah, they had made supersedure cells, but the queen was still in there when I pulled them apart. But it's been 28 days since I saw a capped queen cell, and no signs that any queens made it and came back. So
Yeah, I would recombine them because if you let them go too long, then they might start, developing laying worker issues. It's weird that they didn't requeen.
I also have a fun photo if you want to see. I found our oak leaf maple tree the other day when we walked out to it. I heard before I saw that it was covered with bees, which makes no sense in the fall because there's nothing budding or pollinating. And I looked up to find my bees all over the trees, and they were absolutely going after something, and I couldn't figure it out until I zoomed in really close on the photo, which if you can do that on your screens, you can see that there are little tiny white bugs all over that they're eating.
Yeah,
Are they eating bugs, or is that like honeydew aphids type stuff?
They kind of look like aphids from here.
you see
I'm
how
pretty
that,
sure
that leaf looks all sticky? It looks like it's like got residue, like it, exactly, like sweet exudate, like the sweets, carbohydrate, like liquids that come off of the plant. But Randall and Andy are correct too. Like aphids will often exude what they call it, honeydew.
Yep.
It's like sugary waste product.
They're frass. Yeah.
They're liquid frass. Hey, it's a specialty honey. If you can get enough honeybees to collect the liquid poo of aphids, which is a very sugary substance, it is called honeydew specialty honey, and it's like a food product that's been through the bodies of two insects. The aphid and then the bee, but it's a thicker honey.
I just found that, just Googling it, I found that the Bumblebee Conservation Trust says that they get several calls every year with witnesses saying that they see bumblebees feeding on aphid frass.
They do that too.
Yeah.
And that's a really, that's interesting. This brings up questions that I had with the Kansas group. There's some research out there. Kansas beekeepers have a lot of sorghum around them. And with sorghum is a wind pollinated crop. And so there's fewer pollinator policies and restrictions on that. I mean, it's wind pollinated, so they say it's not attractive to bees. Therefore, there's certain compounds that are allowed to be sprayed that typically wouldn't be. So Foxifer was one of those. That's like the new 4th generation systemic neonicotinoid. They were being used on this product, but because it's wind pollinated, it's not attractive to bees, but they're not considering that they use that product to treat the aphids that are overtaking the crop. And if you have aphids, you have sugar dew. And if it's a systemic product that you're putting on the plant, it's being picked up by the aphids and it's coming out their liquid poo, then it can be picked up by the bees. So there was a researcher out there that was working on sorghum that had noticed a lot of bees foraging out there and was wondering what they were feeding on. But yeah, sugar aphids.
That's funny. And the aphids, for their delicious honeydew, even ants will kind of guard them and herd them around like their own cattle, which I think is pretty fascinating. So yeah, enjoyed by lots of different insects.
My bee mentors told me that bees never come home empty, that they'll either come home with their stomachs full of nectar or full of pollen, and in the ideal world, both. Maybe those bees are going after just the pollen, that they need pollen and they see it there and it's nowhere else.
Yeah.
Or maybe it's good pollen, I don't know.
Oh, in the sorghum.
Oh yeah, it could be. Yeah, I've seen them work ragweed pollen, and that's wind pollinated too.
And corn pollen.
They do, yeah. Yeah, in the spring, sometimes these bees have no pollen sources, and we've seen pictures sent in to us. They're collecting wood dust, because they're so eager to collect pollen, and they're collecting whatever looks resembles pollen. Cool. Well, I guess we are at 6 o'clock. Send us images, pictures, and we will see you next week.
Yeah, thanks for coming, everyone.
Thank you.
Thank you.
Thanks, everybody.
Thanks, guys.
Thank you.